It's wild when you think about it, like all these huge changes to our health sometimes. They can come down to things that we can't even see, these tiny interactions happening inside us all the time.
Right.
We always hear about, diet and exercise, but what if like some of the most basic parts of how our bodies work are actually being set up way back in those first few months of life?
Absolutely.
By like these invisible communities of microbes.
That is exactly the fascinating area we are diving into today.
Okay, cool.
We have been looking at some really, really compelling new research.
Okay.
Particularly a study that was published in Science that kind of goes deep into this unexpected role that microbes early in life play in the development of something super crucial for our health. Insulin-producing cells in our pancreas.
Right. Insulin, that's the hormone that regulates our blood sugar levels, right?
Exactly.
And those are the very cells that are not working properly in diabetes.
Yes.
So the goal of this deep dive is to kind of try and understand new research and what it might mean for how we understand and maybe even treat this really common condition.
Absolutely.
The thing that really kind of hit me right away was the main question the researchers were trying to answer.
Okay.
Because like if our genes play such a big role in type 1 diabetes, why is it that like one identical twin gets it and the other one often doesn't?
Yeah.
They have the same exact DNA.
That's the really critical puzzle, isn't it?
Yeah.
It really suggests that something beyond just our DNA is going on.
Right.
Some environmental factor that is influencing whether someone becomes susceptible to type 1 diabetes.
Okay.
And for years, the scientists behind the study have been looking into the world of microbes for those very clues.
Interesting. Okay, so let's kind of get into the main findings here from this mouse study. What exactly did they find out about the link between early life and those really important insulin-producing beta cells?
Well, the core finding here is really remarkable. They discovered that when they gave these mice what are called broad-spectrum antibiotics during a specific period early on in their lives, this is like kind of a short period. In mouse development, it roughly lines up with about the first 7 to 12 months in a human baby. These mice ended up having fewer of those insulin-producing beta cells in their pancreas when they became adults.
Wow.
They also showed higher levels of sugar in their blood, lower levels of insulin overall, and just generally like poorer metabolic health later on. And just to be clear, those broad-spectrum antibiotics, those are the ones that are designed to kind of just wipe out a wide range of bacteria.
So just to clarify, just a short course of antibiotics early on in life could have these pretty significant and long-lasting effects on their ability to manage blood sugar.
Exactly. It really highlights how sensitive that really early period of development is.
That's a bit concerning.
Yeah, and as one of the study authors actually noted, it was shocking and a bit scary to see just how crucial that community of microbes living in us, what we call the microbiome, is during this like very brief window of time.
Yeah, we've heard before that a baby's microbiome is important, right? Like we've talked about how things like breastfeeding and vaginal birth might be linked to a lower risk of type 1 diabetes.
Exactly.
So how does this new research fit in with what we already knew?
Yeah, we've had hints that that healthy and diverse community of microbes in a baby's gut early on might offer some protection. Right? Breastfeeding and vaginal birth. We know that those influence the types of microbes that a baby is exposed to in those first few days.
Right.
This new study is taking that a step further and trying to pinpoint which specific microbes within that huge community might be the key players in supporting this development of beta cells.
So they weren't just looking at what happens when microbes are reduced by antibiotics. They were actually trying to identify the beneficial ones.
Exactly.
How did they go about figuring that out?
They did a really clever series of experiments. They introduced specific types of microbes to the mice and then really carefully monitored how it affected their beta cell production and their insulin levels.
Got it.
And what's really fascinating is that they identified several microbes that actually increased the production of these crucial cells.
Wow.
And boosted the levels of insulin in their blood.
Oh, that's amazing.
Yeah.
Were there any particular microbes that seemed to have like the biggest impact?
Yes, absolutely.
Okay.
One microbe in particular, a fungus called Candida dubliniensis, showed the most powerful effect in promoting the growth of these beta cells.
A fungus.
A fungus.
That's not usually what I think of.
Right.
When I hear microbiome, I usually think bacteria first.
Yeah, and it really underscores that the microbiome is a complex ecosystem.
Right.
It's not just bacteria. It's fungi, viruses, all these other microorganisms. And this study sheds light on the role of fungi.
Interesting.
Which sometimes don't get as much attention.
So they found this fungus in mice.
Right.
That helped with the beta cell growth.
Right.
How did they then connect that to humans?
Right.
That's where this this poop slushy experiment comes in, right?
That's a very memorable term.
Yeah.
To investigate whether these findings in mice were relevant to human infants.
Okay.
They used fecal samples that had been collected as part of the TEDdy study.
Okay.
The TEDdy study is this huge ongoing research project.
Okay.
Specifically to identify environmental factors that might lead to the development of type 1 diabetes in young people. So what they did is they basically created these poop slushies, which were suspensions of fecal matter, and gave them to mice.
And what did they find out?
Well,
did all the baby poop have the same effect on the mice?
Not at all. It was amazing, actually. When they gave the newborn mice poop samples from healthy human infants within that specific 7 to 12 month age range that we were talking about earlier, the mice experienced a big increase in beta cell growth. So it really mirrored that critical window they had already observed. However, poop samples from infants outside of that age range didn't have that effect.
Really?
And here is where it gets super interesting. They found that Candida dubliniensis was very abundant in the gut microbiomes of human babies. Specifically during the 7 to 12 month window.
Oh, wow.
Suggesting a similar critical period might exist in humans too.
So it seems like there's this window of opportunity in both mice and humans where the presence of this particular fungus might be really important for having healthy insulin production for the rest of their lives.
That's exactly what it points to, this critical period where being colonized by these microbes that promote beta cell growth, like Candida dubliniensis, might be super important for long-term metabolic health.
What happened when they tested this idea in mice that were predisposed to developing diabetes?
Right.
That seems like a really key experiment.
It was a really dramatic result when male mice that were genetically programmed to get type 1 diabetes were colonized with Candida dubliniensis early in life. The number that got the disease dropped hugely.
Wow.
Instead of the 90% of these mice that typically get diabetes, less than 15%.
Wow.
In the group that got the fungus did.
That's huge.
It is.
For one microbe to have such a strong protective effect, it's amazing.
It really makes you think about all the possibilities for prevention.
It does.
And it gets even more interesting. They wanted to see if this fungus could help regenerate damaged insulin-producing cells. So they gave Candida dubliniensis to adult mice that had their beta cells destroyed. And they saw regeneration of those cells.
That's huge.
It is.
If that could be translated to humans, it could revolutionize how we treat diabetes.
Absolutely.
Not just preventing it, but maybe even reversing the damage.
Yeah, it's a very exciting prospect.
Yeah.
But the researchers are really careful to say we need to be cautiously optimistic. They definitely don't want people to take this as a reason to be anti-antibiotics.
Of course, yeah. We definitely don't want people changing their medical treatments.
Exactly.
Based on a study with mice.
Not a good idea.
So what's the long-term vision here?
Yeah.
How could this research lead to actual treatments for humans?
The researchers envision a future where maybe alongside antibiotic treatments, doctors could also give microbe-based drugs or supplements.
Oh, interesting.
That would be designed to kind of protect or replenish these good microbes that might get harmed by the antibiotics. There's also research exploring the use of fecal microbiota transplants for type 2 diabetes, trying to improve metabolic health. But as the researchers point out, that approach has its own risks because a microbe that is beneficial in infancy might not be so good or could even be bad in adults.
So maybe the poop slushy for grown-ups isn't the answer?
Probably not. Given how complex the adult microbiome is, the more promising thing right now seems to be Really figuring out the exact mechanisms that these good microbes like Candida dubliniensis use to encourage the development and regeneration of beta cells.
Got it.
The hope is to kind of use those mechanisms to create new therapies that could help heal a damaged pancreas.
Wow.
And potentially reverse diabetes.
And this research is continuing, right?
It is.
I think I read something about a new facility at CU Boulder.
Yes. The lead researcher on this study, Jennifer Hill, she recently helped to set up a really cutting-edge, germ-free facility at the University of Colorado Boulder.
Wow.
This is a special environment where they can study the infant microbiome in a really controlled way. They can raise animals in totally sterile conditions without any microbes, and then introduce specific ones, one at a time or in combination, to really understand their individual effects and then their effects together.
That's really cool.
This type of research is going to be really important for unraveling all these complex interactions in the microbiome.
It really does seem like how we understand microbes is changing like a lot.
I think so.
We're moving away from just seeing them as these potential threats to actually recognizing all the benefits that a lot of them provide.
Exactly, as one of the researchers pointed out. It's likely that we have far more beneficial microbes living in us than pathogens.
That's kind of a mind-blowing thought.
It is.
When you think about it.
Yeah, and by understanding them better and learning how to use the power of these good microbes, we could find completely new ways to improve and protect human health.
So for you, the listener, the key takeaway from this deep dive is this really surprising and critical role that certain microbes that we meet in those first few months of life appear to play in how our insulin-producing cells develop.
It's a big deal.
This isn't just some abstract scientific finding.
No.
This has really real potential.
Absolutely.
For preventing and maybe even treating diabetes in the future.
We've talked about that critical window during infancy, roughly those first 7 to 12 months, as a really important time for those beneficial microbial interactions.
Right.
And the potential for developing these microbe-based therapies that are kind of inspired by these natural processes offers a really exciting and totally new path for future research and treatment.
It really makes you wonder, doesn't it?
It does.
Given the huge impact of these like invisible microbial interactions early in life, what other things might be going on shaping our long-term health in ways that we're only just beginning to understand?
That's the question.
It's fascinating to think about the interplay between our bodies.
Absolutely.
And the massive microscopic world.
It is incredible.
All around us and inside us.
All the time.
It's a crazy thought.
It is wild.
Thanks for listening.
Thanks for having me.
And we'll catch you next time for another deep dive.
Sounds great.
In the latest scientific research.
Looking forward to it.